Hi, and welcome to the 3P seminar series. This is brought to you by a collaboration between the World Parkinson's Coalition, Cure Parkinson's Trust, and the Van Andel Institute. This week is our last week of our weekly 3P seminars. And from this week on, we'll be going to one session per week, one session per month, with some sessions having a bonus career development or special session included as well. So our first speaker today is Miguel, who is a postdoc at the Van Andel Institute, and he'll be talking to you today about Huntington's disease aggregates and the immune response. So over to you, Miguel.
Thank you, Mickey. Okay, so first of all, I would like to thank Lisa and Mickey for organizing these seminars during these chaotic times. And well, I just want to mention that this is work that I did during a short postdoc in Germany. And it's not over yet, so if someone from the audience would like to collaborate and help me finish this work, that would be fantastic. Feel free to contact me. So, Huntington's disease presents with motor disorders, but also some other disorders like lack of concentration, depression, short-term memory impairments, and ultimately death. It is caused by the loss of medium spiny neurons within the striatum, and unlike Parkinson's, Huntington's disease is completely inherited and it's monogenic. The reason for this is that a mutation in the gene that codes for Huntingtin, and this mutation, what it causes is that it encodes glutamine and causes a polyglutamine tail in the amino terminus of Huntingtin. So if you have 10 to 26 glutamines, it is considered a wild-type gene. If you have 27 to 35 glutamines in your Huntingtin gene, it's considered a risk factor, meaning you're not going to get Huntingtons, but your children may. If you have more than 37 glutamine repeats in the amino terminus, you're going to get Huntingtons. Another characteristic feature of Huntingtons is that mutant Huntingtin creates these inclusions. They are cytosolic and mostly perinuclear. Now, when I arrived to Germany, there was no defined project for me, so I had to find a project for myself. And since I had experience doing siRNA screens during my PhD, I thought, well, I'm just gonna do a screen. I'm gonna knock down genes and see which of these genes upon knockdown causes my cells to have less aggregates, or even better, which of these genes upon knockdown causes my cells to have less aggregates. You can model Huntington's disease by overexpressing the exon 1 of Huntingtin with either 23 glutamines, so that's considered wild type. There's absolutely no aggregates in these cells. Or you can express the exon 1 of Huntingtin with 74 glutamines, and then you start seeing aggregates. So if you're gonna do a screen, what you want is a readout that it can be done high throughput. So the first problem that I had to solve is, how can I tell a machine that this is an aggregate? It's pretty evident under the microscope, but I didn't want to spend hours and hours counting aggregates, especially if you do a big screen. So then I thought, well, within the context of an aggregate, things are in close proximity. So maybe I can get a FRET signal. FRET stands for forced resonance energy transfer, and the principle is quite simple, actually. It means that you can excite one fluorophore with the emission of another fluorophore, if they are in close proximity and if they are in the right orientation. So I already had cells that were stably expressing mutant Huntingtin GFP, and I just threw in an extra construct which was mutant Huntingtin but tagged with M-cherry. So if I tell a microscope excite GFP and read GFP, you see the aggregate, but you also see cytosolic Huntingtin. Well, not cytosolic, but soluble. But if I tell the microscope excite GFP, but this time read M-cherry, what you see is only the aggregates. So now, I started making several cell lines. I made HeLAs, I made HEX, I made U2OS. And out of these cell lines, I started to get clones. So what I noticed was that not all the cells present with aggregates, although it's a clone and everything should, well, all the cells should express the same things to the same extent, not all the cells present with aggregates. However, all of the cells are expressing mutant Huntingtin GFP and also mutant Huntingtin with M-cherry. And the arrow is just pointing at one particular aggregate. So the method worked pretty well. And now if I tell a plate reader, excite 488, but read 630 nanometers, I can read this. So now I can basically count, well, not count, but have a signal towards all the aggregates that I have in that population. So the system was working perfectly and I was ready to do my screen, but then I was still quite baffled about how come some of the cells do not present with aggregates, although it's a clone. So I thought maybe I can separate these two populations, right? So I thought, well, let's throw it into FATS and see how it looks. And this is how it looks. So these are my Fred negative cells. They are only emitting GFP. So GFP goes towards the right. And this is my second population. They are GFP positive. They are towards the right, but they are also up, which is M-cherry, the signal for M-cherry. So after sorting these cells, I put them back into a slide. And you can see that I can have cells that are 100% with aggregates and cells that should have been 100% negative, but keep in mind that this is 24 hours post-sort, so maybe some of these cells started to make an aggregate. Having seen that I can separate these populations, I did just label-free proteomics. I got around 600 genes that were expressed differentially, and when clustered them and asked in which pathways they were involved, I saw that some of them were involved in degradation, so a proteasome, or a viral infection, or a parasitic infection, or antigen presentation, etc. So I started by validating my proteomics results. And this is how the raw data looked on the mass spec. So these are my cells, which have aggregates. These are my cells that do not have aggregates. And as you can see, the results are black and white. It goes from detected to non-detected. All these are subunits of the proteasome. And again, to be sure that they express in Huntingtin to the same extent, I also looked for the peptides for M-cherry, one of my constructs, right? And as you can see, there's no differences between aggregates or no aggregates. So they're expressing Huntingtin to the same extent, but some other subunits are completely expressed differently. Okay, so let's check the proteasome. So via Western blot. What we found is that some subunits of the 26S proteasome, that's the normal proteasome, are expressed to a higher extent in cells with aggregates. Here is Fred positive, Fred negative, or wild type. Some subunits of the immunoproteasome are also expressed to a higher extent in the cells with aggregates compared to no aggregates or compared to wild type. But well, this is not telling you much. This is telling you, yeah, the protein is increased in a population compared to the other, but the proteasome is a huge complex. So does this changes the activity of the proteasome? And the answer is yes. You can measure the activity of the proteasome by using an artificial substrate coupled to a fluorophore. And you can see the orange graph are my cells with aggregates compared to cells without aggregates, the gray one, or wild type cells. Wild type cells. The immunoproteasome changing as well. Yeah, the activity is changing. Again, cells with aggregates, wild type, and no aggregates. Then you can also combine both substrates with equimolar amounts and expect, if you're measuring correctly both types of proteasomes, the normal and the immuno, the slope should be higher. Indeed, it is higher. These are my cells with aggregates. The 7, sorry, I forgot to mention. The 7 just depicts the number of clones that I was using at that time. Another protein that we found, and we found quite interesting, was P62. P62 is the major receptor for ubiquitinated cargo that's got to be degraded via autophagy. And P62 is constantly being degraded, and it should not be there. So we were spotting it in cells with aggregates compared to cells without aggregates. We check for that. This time I'm checking wild type cells, also my clone 7, U2OS, no aggregates, and aggregates, right? And I did a treatment for 0, 5, and 15 hours with rapamycin to this concentration. Rapamycin will trigger autophagy. What we can see is, as I said, wild type it should not be there at all. It's constantly being degraded. But my cells that have no aggregates or less aggregates, keep in mind that all these experiments are 24 hours post-sort. But if you compare it with cells with aggregates, P62 is highly increased at all time points. And also LC32. So this is suggestive of a defect in autophagy. You can also block autophagy, block the acidification of the lysosome and check for P62. This time we're seeing P62 coming in the wild type condition because you're blocking the degradation of P62, but still is far more increased in the cells with aggregates. Then if you want to check for the autophagy flux, what you do is you block autophagy by the use of baphyllomycin 2 hours before, and then you activate autophagy by the use of rapamycin. Again, P62 is highly increased in my cells with aggregates. Another protein that we saw, and it was interesting, was ISG15. ISG15 is a ubiquitin-like modifier and is highly upregulated by interferons. This is the monomeric form of ISG15. These are my cells with aggregates, no aggregates, or wild type. And as I said, ISG15 is ubiquitin-like modifiers, so you are expecting to see a smear. These are proteins that have been ISG-lated. And it is increased in cells with aggregates compared to no aggregates or wild type. As I mentioned, ISG15 is highly upregulated by interferons. So we check for interferons with the RT-PCR. And we found that we have a massive 500-fold increase in interferon alpha for cells with aggregates compared to cells without aggregates, almost nothing for wild type. If you exclude interferon alpha from that graph, you can see the other interferons that I was measuring. Interferon beta is also increased. Interferon gamma is also increased. IL-2. P62 is there because I had seen it being upregulated. So that's just a control. Another class of proteins that we saw changing were those proteins that are code for the MHC1 and the MHC2. Of particular interest were that genes that encode MHC2 were highly upregulated in cells with aggregates compared to no aggregates and compared to wild type. Now, if you have an MHC2 increase, what you would expect is to be able to activate CD4-positive T cells. So that's what we check. MHC2, CD4 recognize each other and activates T cells. So the way you do it, I use URCATs. URCATs are CD4-positive T cells. So I sorted my cells into no aggregates, and then I co-cultivated with URCATs. If a T cell becomes activated, it will release IL-2, and this will cause proliferation. So that's what you measure here. As you can see, cells with aggregates cause a higher activation on these T cells compared to wild type or compared to no aggregates. This time I was using HeLath, but it happens also for U2S. And despite my constant efforts, I was never able to get a clone of this neuronal cell line, SHSY5Y, but I managed to get a stable expressing cell line that expresses mutant Huntingtin. And again, this is significant, at least compared to wild type. And lastly, well, having seen my cells were responding as if they were infected, I wanted to get them killed by immune cells. So this video, the big ones are U2OS without aggregates. The little ones are NKs, natural killers. So the natural killers go into the U2OS, but it's as if they don't know what to do, right? There's even some U2OS proliferating in the top left corner. Now let's see what happened to my cells with aggregate. So they all got killed, strongly suggesting that the immune response that I'm seeing, it's correct. And they got killed in a way consistent with the way NK is killed, which is by releasing granzyme B and perforin. That causes a hole in the membrane, causes an osmotic shock, and that's why you see them exploding. So in summary, I have no idea why some of the cells present with aggregates, some of them not. But we managed to develop a method to separate these two populations, did label-free proteomics. It was indicative of a cell that has been infected. We managed to activate CD4 positive T cells, and more importantly, we managed to kill those cells that have aggregates. With that, I would like to thank my previous lab, the Vilches lab, and my current lab, the Brundin lab. And I would like to thank you for your attention, and I'm open to any question.
Excellent. Thank you, Miguel, for your presentation and joining us today. If anybody has any questions for Miguel, please add them to the Q&A using the feature at the bottom of the screen. So to start off the discussion, Miguel, how do you think that a Huntingtin aggregate is triggering an immune response?
Well, within the aggregate, there's not only protein and there's not only mutant Huntingtin. It is surrounded by several chaperones, and there's also RNA, single-stranded and double-stranded, and that can be sensed by toll-like receptors as a viral infection. Well, that's how I think. I don't have proof of that. I didn't have time to finish that, but that's the way I think it's working.
So kind of following on from that, you're seeing the pathways are affecting different interferons, like the alpha, beta, and gamma. What is triggering the release of each of these different interferons?
Well, again, it depends on the toll-like receptor that you are activating, right? And well, absolutely almost every cell can produce interferon alpha. So it was not that surprising to see interferon alpha going that high. It was surprising looking at interferon gamma. But interferon gamma, it went up, but not as extreme as interferon alpha or beta.
Question here from an anonymous attendee. Are these puncta aggregates? How did you verify these are true insoluble aggregates?
Well, the short answer is no, I didn't verify it, but it is well known in the field of Huntingtons that these are completely insoluble aggregates. Well, maybe not completely, but indeed they are aggregates, but that was well known, so I didn't have to prove it.
I guess a broader question, how do you see these kind of results of the Huntington aggregates stimulating an immune response having an impact on like the clinical condition or the translation? Just how does this research move forward?
Well, I would like to think that there is an autoimmune component in Huntingtons that has been overlooked. Most of the research in Huntingtons like focused on looking at the content of the aggregate. What we did was looking at the cellular response towards the aggregate. So, and if interferon gamma is indeed released, it will permeabilize the blood-brain barrier. So potentially leading to infiltration of immune cells and probably killing neurons.
So you think maybe by combating the immune response, that could be a potential avenue for therapeutics for this disease?
I would like to think
hypothetical
so.
at this point.
I mean, you may not cure it entirely, but it may delay, but it may enhance your quality of life, right? I would like to think so. I couldn't prove it, but yeah.
And do you think that this is specific to the Huntington aggregate, or do you think other neurodegenerative diseases like Parkinson's and Alzheimer's that is also having this aggregation of proteins would have a similar response?
My idea is that maybe different aggregates trigger different responses, but I think personally that they all will cause an immune response at some extent, to some extent.
Yeah. Excellent. Are you planning to do any RNA-seq on your second segregated populations? Question from Sandra.
Well, the thing is that I'm no longer in that laboratory, right? So I actually couldn't finish the whole thing. It would have been interesting to do RNA-seq, but in my case, proteomics was the fastest way to have a quick look. But I mean, of course, it would be nice to have to support those results with an RNA-seq assay.
And a question here from Kurt. Would inhibition of proteasome function itself make a cell target for natural killer cells?
I'm not sure about that because, well, it had been proposed that Huntingtin aggregates were inhibiting the proteasome activity, but that's not what I saw. What I saw is that my cells with aggregates had a much higher activity, proteasomal activity, but they also have an impaired autophagy. So I'm not sure. I'm not sure about that. But yeah.
Okay.